Mr. V's Biology.
Science rules.
Mr. V's Biology.
Last page. Complete the chart below. Macromolecules. I have carbs, lipids, protein, and nucleic acid. The function of carbs is the main source of energy. Lipids is the long-term energy storage. Fat. Protein. Make stuff. And looks like we might have to move that down. Nucleic acid stores the genetic information. Stores genetic information. The monomer, what makes them up? So carbs are made up of monosaccharides. Lipids are made of glycerol and fatty acids. Protein is made of amino acids. And nucleic acids are made of nucleotides. So examples of carbs: bread, pasta, potatoes. So that's sugars and starch. So fruit, veggies. Lipids would be oil, fat, and wax. Protein, meat, nuts, beans. Nucleic acid, DNA, and RNA. We're just going to talk about number 9. What is a chemical reaction? Well, that's just a reaction that takes place where two or more chemicals react with each other and make a product. So the product is what is made. So I'm going to label the following. I know that B is the enzyme. A is the substrate. That's what the enzyme is going to go bond to. C is the enzyme substrate complex. E is the product. That's what's made. And I know that D here is the active site. This one here is just the enzyme, ready to go and find another substrate and do the whole thing all over again. All right, now we're down here. A couple more to go. What is the role of enzymes in biological systems? So why do we need to learn about enzymes in biology? Is because they speed up chemical reactions. So that we can live. The diagram below represents a human enzyme and 4 types of molecules present in a solution in a flask. Which molecule would most likely react to the enzyme? So here's the enzyme. Which of these molecules would react to it? So if I look at the active site and I look at these substrates, I know that it's going to react with letter D. Explain why. The active sites are the same shape. Use what you know to match the enzymes with their substrates and functions. So we haven't talked about these, but you can apply what you have learned to figure out what they do. So amylase, I know, is an enzyme because it ends in that ASE. So which of these would be the substrate. Synthesizes DNA, digests sugar, maltose, digests starch, amylose. Oh, so amylose is the substrate for amylase. So I know that this is number 3. Protease digests proteins. Lactase digests milk sugar, which is called lactose. DNA polymerase works with DNA, synthesizes DNA. 1. Maltase digests sugar in beer, which is maltose. And ATP synthase synthesizes ATP. Number 4. Awesome. So you are done with your packet. Most everything in Unit 2 biochemistry packet should be complete, except for a couple labs and such that we weren't able to do this year. Now it is time to play a review activity to get ready for the test.